I don't like this. I don't like this one bit. This is our project, the Nutritiousness of Photosynthesis. Let's go ahead and take a journey into the chloroplast. We are now inside of the chloroplast, looking at the thylakoid membrane, represented by an orange peel. On the membrane, there are some proteins that we need to talk about. We have photosystem 2, PQ, the B6F complex, PC, photosystem 1, FD, NADP reductase, and the ATP synthase. When sunlight strikes photosystem 2, H2O, which is broken up into electrons, hydrogen ions, and O2 gas. The electrons move into photosystem 2, while the H+ ions are spread in the inside of the thylakoid membrane, along with the oxygen gas. Now, in order to move the excited electron through the thylakoid membrane, we use an H+ ion to activate the PQ protein, allowing the excited electron to be moved to the B6F protein. Once the electron is there, the hydrogen ion is then moved into the inside of the thylakoid membrane. With light striking photosystem 1, the excited electron is then moved from the B6F complex to the PC protein and then to photosystem 1. The excited electron in photosystem 1, with light striking it, is then moved to the FD protein and through the NADP reductase protein, where NADP+ is reduced to NADPH. Now, looking at ATP synthase, due to the concentration gradient of hydrogen ions on the inside of the thylakoid membrane, hydrogen protons diffuse through the ATP synthase, causing it to spin and attach a phosphate to ADP, forming ATP. We are now in the stroma of the chloroplast, where the Calvin cycle occurs. The Calvin cycle is made up of 3 parts: carboxylation, reduction, and regeneration. In the first step, carboxylation, CO2 is added to a ring of 5 carbons known as RUBP. This reaction is catalyzed by an enzyme called rubisco. This makes an unstable 6-carbon compound that then splits into 2 molecules of 3-PGA. The next step of the Calvin cycle is reduction. In reduction, ATP adds a phosphate to the 3-PGA and is broken down into ADP. Then, hydrogen and energy through electrons are added to the ring of carbon by the use of NADPH, breaking into NADP+ and protons. Next, after the oxidation of NADPH into NADP+, 3-PGA is converted into GAP. Then, by the enzyme phosphate isomerase, GAP is interconverted into DHAP. Together, they are referred to as triose phosphates. 1 of every 6 molecules of triose phosphate leaves the chloroplast to become a larger carbohydrate like glucose. Next, the remaining triose phosphates, with the use of ATP, are used to regenerate RUBP. This happens to 5 out of 6 triose phosphate molecules that are made to ensure the process continues. Next, NADPH is the brightness went down, and then this thing popped up.